Well, here we are again. It's hard to believe another day off. Um, so I'm going to just narrate half of the DNA PowerPoint, Chapter 16. And again, the goal is that Friday this week, Dr. Hayes and I will be available to go over material that you missed, that you viewed already online, and anything that you have questions about. So take some notes about some questions that you have. Okay, so we've learned a lot about mitosis, meiosis, and genetics, and now we're actually going to just talk about more that the some historical information about how scientists figured out what DNA was, or what the genetic material was, I should say, that it was DNA, not protein, for example, and also how DNA replicated and passed on the genetic information. So that was something that got discovered as new technology came into play. All right, so you probably have seen these gentlemen, Watson and Crick, who discovered the structure of DNA in 1953. I know that doesn't sound like a long, maybe to you it sounds like a long time ago, but in terms of just science history, it's not that long ago. All right, so this was just the article in the paper describing, or I guess in the science journal, describing what was discovered. And we're going to see that other folks helped them come to the discovery of the DNA being a double helix. Anyway, the next couple of slides just give us a little bit of the objectives for this particular chapter. And again, some of this is historical, and just to give you some background, and then the rest is just kind of getting you up to speed with what is DNA made of, and what's its structure, and how is it replicated. Okay, so that's some of the overview questions we'll talk about. So again, the question was, how was the genetic material discovered? Various scientists contributed to this. Early scientists in the 1800s, Weismann and Nageli, said it was within living cells, the genetic material, it has to be passed on. So they knew it's something that had to go from parent cells to daughter cells. They found some chromosomes. They thought they didn't know what they were, but they called them colored bodies. And they became convinced that those were the genetic material. But the question really was it protein or DNA? So that was a big question at the time. The DNA or the protein, what was the genetic material? What was the hereditary material? Let me go back. I'm trying to do the laser. It's its own challenge here. Okay. Oops. Here we go. Okay. And so the strain labeled smooth, which had a capsule around it, so the bacterial cell, picture this, had a capsule around it, that bacteria was very dangerous and fatal to mice. Okay, so the one with the capsule, called smooth, would get into mice and kill them. The other bacterial strain, called rough, lacked a capsule, and so when you put that into the mice, it was not fatal. Okay. It's thought that the capsule protected them to the bacteria so that the mouse's immune system couldn't attack the bacteria. So that's why it was fatal in mice. But without the capsule, the bacteria was vulnerable and got killed by the mice immune system. Okay, so you just have to know smooth is fatal in mice, rough is not fatal. Okay, and so he did a bunch of different variations on an experiment to see, what happens when you inject the bacteria into the mice, the different strains. What happens if you decide to, let's say, take the bacteria and heat it, kill it, or inject it live and things like that. And so what he found was that when we know that, S will be fatal in mice and R, rough, will be not fatal. But what happens if you heat kill the S? Well, now the mice survived. That was different, right? So if you actually heat the S up before you put it in the mice will survive. However, if you take that heat-killed S, which causes the mice to survive, but you add live R, which normally does not kill mice, the combination of those two will actually cause the mice to die. That was a huge discovery. That was some kind of transformation. They used the term transformation. So something transformed the normally benign type S. I'm sorry, the normally something transformed or left the R strain and went into the S to kind of make it kill the mice. So it looks like there was definitely some kind of transfer of genetic material. Okay, so again, this is just a picture of it, but again, S is normally kills the mice. So living S, put it in, kills the mouse. R normally does not kill the mice. Mouse is healthy. If you heat kill the S, then the mice live, so you don't kill the mice. However, if you heat kill the S but add some of the R, something causes the mice to now die. And basically, something from the R transformed the bacteria to become living S, or dead S, and that will then cause the mice to die. So again, the big thing was this phenomenon is called transformation, which is a change in genotype and phenotype due to assimilation of foreign DNA. So there was some kind of exchange of DNA, transfer of DNA from one strain to another. So that was huge. No one knew that that could happen, that DNA could transform or exchange between two different strains. So that was a big thing. However, despite that exciting news, other scientists didn't know, was the transforming substance DNA or proteins? So that was still a burning question. Was it a protein that was doing all this transformation, or was it DNA? Okay, so like we said, they wanted to figure out what the genetic material was made of, DNA or proteins. So other scientists checked this out, and what they did was they kind of repeated the experiment, but changed a few things during the transformation. So like if they did the same experiment, but included DNase, RNase, or protease, what would happen? So remember, ASE at the end of a word means enzyme. So let's see, first, for example, if they did the transformation and had RNase present, that would degrade or break down RNA. And so if RNA was the transforming substance, then the transformation wouldn't work if RNase was present, because RNA would then be destroyed. But when they used RNase, the transformation still worked. Likewise, protease, that's an enzyme that degrades, breaks down protein. If protein is the transforming substance, and you add this protease enzyme, and you still get transformation. Oh, I'm sorry. If you think protein is the transforming substance, if you added the protease, then that would degrade the protein, so then you wouldn't get transformation. However, they found that when they add protease to the transformation experiment, transformation still worked. So that told them that protein was probably not the transforming substance. I hope everybody's catching on with this, right? Finally, they used DNase with the transformation. So that's an enzyme that will break down, degrade DNA, cut it up. And when they used DNase solution, lo and behold, the transformation did not work. That tells them that DNA must be the genetic material. Okay, so the take-home message is that DNA is the genetic material. Griffin found that there is a transforming ability with this genetic material, and others after him, like Avery, McLeod, and McCarty, determined that DNA is what is actually the genetic material, not protein. Okay, so the next uh, bit of information is a little bit more about the structure. And we're going to look at levels of structure of DNA. And so, we know the building blocks are nucleotides. We're going to study those, and then we're going to look at a DNA strand, a double helix, formation into the chromosomes, and finally, the word genome just means all the DNA in an organism or species. Okay, so we're going to start with first the nucleotides. So you learned this earlier on in the semester, so we don't need to spend a lot of time on this, but nucleic acids is a category of what you find for DNA, and that's a type of macromolecule. And the building blocks are the nucleotides, which contain a phosphate, a sugar, a 5-carbon sugar called pentose, and a nitrogenous base, meaning containing nitrogen. Okay, here's the structure of a nucleotide. Again, a phosphate, a sugar. In the case of DNA, it's deoxyribose. I just want to highlight that you see this 2' position here. So the carbons are numbered this way of the sugar here. So you have like carbon number 1, 2, 3, 4, and then this is the 5th carbon right up here. So carbon number 2 does not have an oxygen. You see it here on number 3. So this is lacking in oxygen, so that's why this sugar is called deoxyribose. Okay. Also note important things like said over here. The base is attached to carbon 1 off the sugar, and the phosphate's attached to carbon 5 off the sugar. Okay, that's the traditional numbering system. Here we see it again. And I guess the other thing just to mention is that the bases, there are 2 kind of groupings of the bases: purines, which actually have 2 rings, and pyrimidines, which are a single ring. And the way I remember that is that purine is a smaller word, but it has 2 rings. It's kind of ironic, right? And pyrimidine is a bigger word, but it only has 1 ring. That just helps me. Okay, so the purines also are the A and G bases, adenine and guanine, and the pyrimidines are the T and C, thymine and cytosine. Okay, now what we're going to do now is contrast that to RNA, the nucleic acid RNA. And so in terms of the building blocks of RNA, you still have a phosphate and a sugar and a base. The sugar is called ribose, not deoxyribose. Note that you see at the 2' position you actually have an oxygen. So it's not lacking in oxygen. That's why we don't call it deoxyribose. Instead, we call it ribose. The other difference here is that the bases are very much the same except for here. One of the pyrimidines we do not have thymine. Instead, we have uracil. So we still have the adenine and guanine and cytosine, but instead of thymine, we have uracil for RNA. Okay, looking at just kind of how the nucleotides build a strand, you can see that what's going to happen is that we're going to have covalent bonds between the nucleotides. So, for example, we have our phosphate sugar base, and then we're going to connect that to another nucleotide through the phosphate. See the phosphate? So the phosphate links two sugars, as shown here. Okay. You can see it's going from the 3' carbon at one sugar to the 5' at another. The 3' there. Okay. So you can kind of see where that comes from. So the phosphate groups link the two sugars, and they call these bonds phosphodiester bonds. Okay, so again, those bonds that link the two sugars together through the phosphate are called phosphodiester bonds. Okay. The other important thing to know is the bases are in the middle, so we'll see how they link together with the other strand. But right now this is just a single strand, what you're looking through here. And notice there is a directionality to this. So there's a free end here that is not linked to any other nucleotide, and that's called the 5' end. So that end is not linked to any other nucleotide. The other end of this strand is called the 3' end, and that also is free, and that's the OH. Basically, the OH at the 3' is not linked to anything else. So at the 5' end, you have the phosphate of the strand, and the 3' OH is free, and there's nothing attached to that. And so we'll see actually that when we build a strand of DNA, we're actually going to do it in one direction only, meaning we're going to add to the 3' end. So we're going to see that coming up. But basically, if you were to look at the sequence of this strand, you would name it as 5' to 3'. So you would say 5', like it's here, T, G, C, A, 3'. Okay, so you definitely need to say which direction is which strand is which, etc. This is special orientation for DNA. Okay, we're going to move a little bit more now into structure, how this all was figured out. So Watson and Crick wanted to know the structure of DNA, but they were not alone. This woman here, you may have heard of her, Rosalind Franklin, was actually the work behind all of this interpretation. So she did the actual experimentation. Also Maurice Wilkins too, using X-ray crystallography to study the DNA to figure out the structure. And so Watson and Crick kind of interpreted her results, but she did the experiment. She did the work. Unfortunately, she didn't get much credit until after she died, because Watson and Crick won the Nobel Prize. She didn't. But when she did the work, they found this X shape to the DNA, to the double helix. So that spacing and the presence of an X suggested there were two strands. And it also suggested a helical structure. And the spacing was such that it suggested that a purine was placed with a pyrimidine when they matched up, since the size were different. Remember, the purine has 2 rings, the pyrimidine has 1. So it's not like two purines got together or two pyrimidines, but 1 and 1 seemed to be the best space. And how they figured all this out was this technique called X-ray diffraction, which was a new technology that was available, which allowed them to make this discovery. What they did was they took a piece of DNA fiber and they focused an X-ray beam through it. When it went through the DNA, what would happen would be the X-rays then would scatter based on the atomic structure of the DNA. And when those X-ray beams diffracted, they call it diffracted or scattered, it was detectable on a piece of film, and then you could develop the film, and what they noticed again was this X shape, and the spacing was critical. So they were able to determine that DNA is a double helix, and that a purine will pair with a pyrimidine. Now, what else do we know about the pairing of the bases? Well, Chargaff in 1950, around the same time that this was, actually a little earlier before this was found, did a whole bunch of experiments looking at different organisms, anywhere from bacteria to chicken to salmon, human, etc., and compared the amount of adenine, the amount of thymine, the amount of guanine, and the amount of cytosine. Okay, as you can kind of see here. And what he found consistently through several experiments was that the amount of A, or adenine, always equaled the amount of thymine. So you can kind of see that here. Here in yeast, you have 31.7 and 32, which is roughly the same thing. Human, you have 30-30. E. coli, 26-23, very close. And the amount of G always equaled the amount of C. So here we have, 22, 21, 19, 19. So, it didn't matter what species you were talking about. A always equal to T and G always equal to C. Okay, so actually that work supported the idea that there were two strands and that there was this pairing, complementary base pairing between A and T and G and C. So Watson and Crick brought all this information together. So they got the information from Rosalind Franklin as well as Chargaff's information and found that there needed to be a double helix. The bases had to be in the center. A and T and C and G were the only way to fit it. And so this was published in the Nature magazine, which is a very respected magazine, in 1953. And they won the Nobel Prize with Wilkins in 1962. Unfortunately, Rosalind Franklin, did not get put on that paper, or that, I'm sorry, she didn't get published. Sorry, she did not get the Nobel Prize with them. She was not included in that, even though she did the work, which was not very good. So she should have gotten more credit than she did. In her obituary, someone wrote, "Her photographs are among the most beautiful X-ray photographs of any substance ever taken." So there's actually now a medical school in her honor, in her name. So she did get eventually, did get recognized for her extraordinary work. Unfortunately, it was after. Okay. Back to the structure. So they were able to figure out that this is this double helical structure. Bases on the inside. Sugar phosphate backbone. We saw that before. And we know too now that the bases are held together, AT or CG, by hydrogen bonds. Okay. A little bit more. Just some details that there are 10 base pairs per turn in the helix. double helix, that the two strands are complementary, which means we said G and C go together and A and T go together. However, notice the way they're written. So when we talked about 5 prime, and then we have our sequence 3 prime, that's the other end. Notice that when those two strands are together, they're actually running in different directions. So here is the 5 prime end with the free phosphate, and here's the 3 prime end with the free OH, and on the other side you have the free 5 prime end with the phosphate, and here you have the free 3 prime end with the OH. So they are running what we call anti-parallel. So they are not in the same direction, they are anti-parallel. So one strand is running 5 prime to 3 prime, the other strand that's matching with it is 3 prime to 5 prime. And they found that that must be the way they fit together to best fit the spacing for the double helix. So here's a question for you to ponder at your places where you are. What would be complementary for the following? So for example, here's our 5 prime TAC GT 3 prime. So when you answer the question, you have to write the complementary base pair as well as the orientation of the 5 prime 3 prime 3 prime 5 prime. Same thing here, except of course it's a longer strand. Okay, so give yourself a minute to scribble down the answers, and when you're ready you can go to the next slide, and I'll give you a second. Okay, hopefully your answers match up with mine in red. So we've written the complementary strand. For example, what goes with T, A, what goes with C, G, etc. And notice it's the opposite orientation, so the top strand is 5 prime to 3 prime, the bottom strand is 3 prime to 5 prime, and the same exact thing is happening down here. Okay, sometimes you'll see DNA displayed in a variety of ways. There's one type of structure called the space-filled model, where you can kind of see how it's, you know, they've kind of filled up all the structures so that you can actually see the realistic kind of view of this and the bonds and whatnot, and actually the atoms and things. And what you can see is that when you look at the DNA, there's actually these little grooves of how it twists. And so you'll have like this huge space here, then a little one, and then a huge space here, and then a little one every time it turns. And so there's something especially I'll point out called the major groove, which is an important place where proteins bind to the DNA and affect gene expression. And we will talk about that in the future classes. Okay. But just to bring up the idea that DNA is going to be interacting a lot with protein. Okay, so now that we know the structure, they've accepted the structure. At the same time, they were also trying to understand, how does DNA replicate? Because we know it replicates, right? We studied that in cell cycle. We looked at it in interphase and the importance of replication of DNA, chromosomes, etc. So there were 3 models proposed to explain how DNA might be replicated. And they were called semi-conservative, conservative, or dispersive. Do you know what the accepted model was? Well, the answer is semi-conservative. Okay, we're going to take a look at what that means. So DNA is replicated in a semi-conservative way. Okay, so we're going to look at what that means in terms of the parental strands and the daughter strands. So I use those terms to explain different things. The parental strand is the original strand of DNA. and a daughter strand is the strand that's made during replication. Okay, so just get that straight. So the parental strand is the original strand of DNA that you're replicating, you're using to replicate, and then the daughter strand is the new strand that you're making. Okay, so just in a picture, this is what some differences of the semi-conservative, conservative, semi-conservative, and dispersive. So in conservative, you're kind of keeping the parental strands, for example, and conserving them so that they're intact. In semi-conservative, we'll see you're going to use one strand and keep one strand and make one new strand. And dispersive is just a mess, just like a little bit of both things. So what we actually are going to see is, like I said, it's a semi-conservative type of DNA replication. And so in 1958, so that Watson and Crick published their thing in 1953. This is a few years later. 1958, Messelson and Stahl, who are shown in the picture, buddies, wanted to find the correct mechanism for replication of DNA. And they used also bacteria. Again, bacteria is a real friend to the scientist. Besides making us sick in many ways, we can use them to study a lot of different things. So they used E. coli. You've heard of E. coli. E. coli, and we know that nitrogen can come in isotopes, so that we can have an isotope of nitrogen. And so we can have what we call light nitrogen, nitrogen 14, which is considered light, and then another extra neutron in there would be heavy nitrogen, nitrogen 15. So what happened was they decided to grow E. coli in nitrogen 15 medium, which is heavy. So the DNA, as it was being made, it needs nitrogen. Remember, nitrogen bases. So the DNA became heavy and labeled with the N15. After a certain amount of time, they put the E. coli into a new medium that contained N14 medium, which was lighter. And then they collected samples to see what does the DNA look like now. So what they saw was that the parental pieces, meaning the original pieces that were starting out, were N15, because that was how the DNA was grown in the beginning. And then when they switched it to N14, new strands that were made were N14. So that helped explain that there was this semi-conservative mechanism going on where, as you can see here, at first everything is N15 in red. And then when they switched it to N14, what had happened is this DNA separated, and one parent strand was used to make a new strand, which was blue, and the other parental strand was used to make a blue strand, which was new. And so what they did was, after they did the switch to N14, they actually got like a hybrid strand, which was a mix between N14 and N15 in terms of density. What this on the right-hand side is a centrifuge tube and a gradient where this bacteria was separated based on density. And we know that N15 is heavier than N14, but a mixture of it together would be considered what we call hybrid. So by going from here, N15, to this first generation, we see that the DNA that's made incorporates a new strand. It uses an old strand and makes a new strand, and uses an old strand and makes a new strand. So it's a hybrid of both strands after the first generation. After the second generation, then it's different. But after the first generation, you can kind of get the information that it's definitely semi-conservative replication. I hope that helps. I hope you understand that. That's just a little way of figuring that out using these different isotopes of nitrogen. Let me know if you have any question about that. Okay. So the take-home message is that the parental strands, which are the original two strands of DNA, serves as a template to make daughter strands, which are the new strands. And when you do this, you follow the complementary base pairing. A goes with T, C goes with G. And the end result is that you get two double helices that develop, each of which has one old and one new strand, one old and one new strand. Okay, so you can kind of see that here. This is from your book. Here's the parental molecule again, which is the parents. Once those split, here they're split. So they're the original. And what you make from that is that you then pair up and use new DNA and you match up to make complementary DNA on this strand and complementary to this strand. And so in the lighter blue, if you can see that, those are the daughter. That's the daughter strand there, and that's the daughter strand there. So the consequence is that the DNA that arises here after replication is a combination of one old and one new strand, one old and one new strand. Okay, and notice the anti-parallel nature. Okay, and we call this semi-conservative. Okay, here's another picture in case this helps. You have your DNA. These are the parents, the original strand. It separates. And each parental strand, the red, is used as a template to make a new strand, which is in blue, the daughter strand there. So then you can kind of see the products of replication are one old, one new, one old, one new. I think you get it now, right? Good. Okay, here we are. Now, what we have next is a look at the details of how DNA replicates. So what we have to discover is what are the important parts of replication, how does it get started, and what proteins, enzymes, things like that allow for replication. Now we're going to mostly concentrate on eukaryotic replication. Prokaryotic is much simpler, but there's similarity, but we're mostly going to talk about eukaryotic. So, DNA replication, how does it start? It starts at the origin of replication, right? The origin, that's where the beginning is, right? So that's why they name it the origin of replication. And what that is a sequence that is the start site for replication. So it's a sequence on the DNA where it will open up and unwind with the help of proteins. But that's going to be the place that says this is where to start replicating. This is getting cut off for some reason, but bacteria have only a single origin of replication. And they have usually a circle, that their DNA is in a circle. Whereas eukaryotes have generally linear DNA, like not in a circle, and eukaryotes have multiple origins, whereas bacteria have a single origin of replication. So when the origin of replication is identified, and what will happen then, we'll see, is that at the origin, as the DNA unwinds a bit, you form this replication bubble, and there's a Y-shaped part to this, as you can see, on each side, and so that is called a replication fork. This sort of Y-shaped area around the bubble, that's where new DNA strands are elongating. Okay, so we're gonna spend a lot of time on this. Now, here's my friends. You have several key players in DNA replication. Not sure where we found all this, but these are our friends that help this whole process. So if this helps you, hopefully, remember, then let's do it. So we're going to see a bunch of friends: helicase, DNA polymerase, enzyme, primase, ligase. Notice they all have ASE, 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 ASE. They're all enzymes. We're going to see. Okay, so first our friend helicase, DNA helicase. So DNA helicase is an enzyme that will bind to DNA. So here it is. And it will travel 5' to 3' using ATP to separate the strand and move fork forward. Okay, so it's going to help basically separate the strands, unwind the strands at the fork there, and it needs some energy to do it. Okay, so that's necessary because you can't replicate the DNA if it's all wound up tight. Okay, so we have our helicase. There's another enzyme which wasn't listed before, but it's called DNA topoisomerase. It's shown here. This actually often will travel ahead of the helix where you're actually replicating to help with the unwinding or the overwinding of the DNA. Because picture like if you have a slinky, and if you ever tried to separate the strands of a slinky, it's kind of like that. When the DNA unravels a little bit and forms this bubble and Y-shaped fork, that's going to cause some downstream overcoiling of the DNA and twisting. And so there's this enzyme, topoisomerase, that kind of relieves some of the strain from the over-twisting. And so there's a lot of details about that. I don't really think you need to know that yet, but just realize that there's an enzyme that will help with that. There's also these single strand binding proteins that keep the parent strands open so that they don't close back and rewind together, which would prevent replication. Okay, so those are some of the first few things to look at. Next, we have our DNA polymerase enzyme. Now, there are some complications that there's different kinds of polymerases, but for our purposes, just call it DNA polymerase. Doesn't matter if it's 1 or 2 or 3 or whatever. That is an enzyme that will covalently link the nucleotides together. And here it is. It's huge. It's an amazing complex, and it's going to help provide the place for the incoming nucleotides, deoxynucleotides, to bind and find their partner on the template strand. But one thing to note is that DNA polymerase is great, but it's unable to build or begin DNA replication on a bare template strand. So, for example, it just can't start making DNA out of nothing. It actually needs to build onto a 3' OH end. So the step before actual DNA synthesis is that there's an enzyme called primase, shown here, which will make a short RNA primer. And so this enzyme primase will construct a short RNA primer, shown here in yellow, which will provide a free 3' OH end to which the DNA polymerase can bind and start building DNA. So that's a necessary step. Okay, so a primer, RNA primer, and after the primer is made by primase, then DNA polymerase will bring deoxynucleotide triphosphates and match up the template strand and build a new strand from that. But again, building onto the primer. So here's the primer in yellow, and here's the DNA that's now been built. Now notice, DNA polymerase can only synthesize DNA in the 5' to 3' direction. That is true. only in the 5' to 3' direction. There's what we call a leading strand, which is made in the direction of the fork opening. So the leading strand is a new piece of DNA that is synthesized in the direction of the replication fork opening. So you can kind of see that here. If the fork would be opening, this DNA is synthesized in the direction of the fork opening. We call that the leading strand. That's made after there's a little primer. And again, this is in the 5' to 3' direction. There's no numbering right on here, but that's what's supposed to be the 5' to 3' direction. And here we can kind of see it in more detail. So the fork is open. These are the parental strands. And what's growing here is the new strand, daughter strand. Here's the primer in the orange, RNA primer. Notice it's 5' to 3', and here's the top strand, 5' to 3' up here. So here's the 3. So you have to show that the 5 is here, because remember it's anti-parallel. So here's the primer, and now you're building DNA onto the RNA primer. Here's the 3' end of the primer. So you're building onto the 3' end. Here's the continuous piece. They call this the leading strand. It's made in the direction of the fork opening. You can kind of see the fork unraveling here. So it's called continuous replication, and it's the leading strand made in the direction of the fork opening. Is that okay? So the direction of the fork opening, you're making the leading strand. It's continuously made, meaning in one piece. You have one primer, one piece, and as it opens, it just keeps being made. The other hand, you have what we call the lagging strand, which is going in the opposite direction, okay, away from the fork opening, okay. So again, it's still going 5' to 3', but it's going in the direction opposite the fork opening, since they are different, because remember, they're anti-parallel. They can't be made in the same exact way. The lagging strand is made in pieces, discontinuously, not continuously. And the pieces are actually called Okazaki fragments, named by the founder of this, Okazaki. And so we're still synthesizing the DNA 5' to 3'. We're using primers, but here you actually have to use a lot of primers, because every time, so here's a primer in orange, and then you build a little piece. And then as the fork opens behind you, you build another primer and another piece of DNA, and then a primer and a piece of DNA, and then a primer, a piece of DNA, a primer, a piece of DNA. Because as the fork is opening behind you, then you're going to replicate by backing up and then replicating, backing up and then replicating. So it's different. Here you can see it in a hopefully a clear way. So in this case, here's the blue are the parental, the red is the new strand. So what we saw before was the leading strand, for example. We have our primer, and we're opening the fork in this direction, and so the leading strand was made continuously in one piece in the direction of the fork opening. The lagging strand, on the other hand, is made in the direction opposite the fork opening. So the fork's opening here, this is still made in this direction. 5' to 3'. First you build a little RNA primer, and then you make the DNA. And now, if you can imagine this opening, and then you're going to make a primer and DNA, and a primer and DNA, and primer DNA. So you have to back up. So lagging strands are made in the direction opposite of the fork opening, only made in the 5' to 3' direction. A new primer is used each time. Now you have, with the lagging strand, all these pieces of DNA, so really built in pieces, but also it's a combination of DNA and RNA, right, with the primase and everything. So what's going to happen is at the end of replication, the RNA pieces are going to be removed by the DNA enzyme. DNA will fill in and replace that stuff with DNA. And then DNA ligase, the enzyme, will catalyze the linkages between all the pieces of DNA. Okay, so you're going to replace the RNA with DNA using DNA polymerase, and then DNA ligase, well, basically it's like tape or glue, will seal the pieces together of the DNA. Now, what I have here, I have a worksheet I will give you whenever I do see you, when the snow is over, and you can actually practice drawing out the leading strand and the lagging strand. And so what I've given you, and again, I have this on a piece of paper, but if you want to see this, what's going to be, this here is the parental strand, and this one is also the parental strand. So I've drawn basically if you can picture, the rest of the DNA would be over here and over here. So this is like the replication bubble. Here's the fork. So that's the fork here and the fork here, which is the Y-shaped part of this. And so the fork is opening this way, so it's going to continue to open. So you want to make DNA that's leading in the direction of the fork opening and then lagging, which is made in the opposite of the direction of the fork opening. So what you could practice doing is kind of drawing in your own leading and lagging strands using my parental DNA right there. See if you understand. This is also a nice summary of DNA replication photo here, where you can look at all the different players, the helicase, the binding proteins, the leading, the lagging, the primase, the polymerase, the overall direction of replication. So, you know, there's different ways that this is often drawn in books. You do have to just be aware of the orientation, the 5', the 3', etc., but also realize all the important parts and all the important proteins that help carry this out. And then I encourage you to watch this animation. Okay. So, DNA replication is very accurate. So why is that? Well, it seems that the stability of matching A and T and C and G is there. That there's hydrogen bonds allow for a lot of stability between A and T versus C and G, but any other combination wouldn't be as stable. Also, the DNA polymerase enzyme complex, the active site is best fit when the proper bases are bonding together and matching together. So it's encouraging the right partners to get together. And also, the DNA polymerase enzyme actually can proofread and look at the pairing of the bases, and if there's any mistakes, it can kind of back up and replace to digest away the mistakes and replace it with the correct base pairings. So it's part of the repair process. So, I mean, of course, there are still mutations that occur and problems, but what this is showing is that God designed it so that you have some built-in safeguards. Okay, so despite the good ability of the DNA to proofread and, you know, pair properly, that's all good. There is one area of replication that can be tricky, and that's replicating the ends of DNA. That when one of the ends has difficulty in being replicated, because as you're trying to back up, especially the lagging ends, it's hard to back up and replicate the end when there's no place to copy and no place to back up on. So repeated rounds of replication at the very tips can lead to shortening of the DNA molecules. The prokaryotes, there's not a problem because it's usually circular, but eukaryotes, because it's linear, the ends may start getting shorter and shorter. And so the solution for this, at least partial, is what we call telomeres. These are a series of short nucleotide sequences that are repeated at the ends of eukaryotic chromosomes. And so that, for example, here, these sequences are called telomeres. These are repeated sequences which help extend the chromosome a bit so that they form with this 3' overhang so that it can provide a place then for more DNA to be created at the end of the chromosome, place to kind of come from. And so there's an enzyme called telomerase. Telomerase. It's a very important complex, very important enzyme. And what it can do, it'll basically put on, it's an enzyme that can do two things. It can kind of put on the DNA sequence, okay, at the end of the chromosome, and then fill in, the DNA for the other strand too. So it's a really good enzyme. It'll put on the repeated sequences and help build the strand of DNA and lengthen the chromosome. And so the problem is telomerase can decrease over time as you age. So there's a lot of interest in understanding the use of that enzyme and how it actually declines over time. So as you get older, your chromosomes may start shrinking because telomerase will not function as well. On the other hand, they've seen that telomerase enzyme has actually been high in terms of high levels in cancer cells, so that cancer cells can grow well and replicate their chromosomes, and telomerase is very active, and so that's kind of an indication of difficulty there with cancer cells. So if you're ever interested in researching any topics, telomerase enzymes, it's a very interesting topic because, like I said, with aging they see a decline, with cancer they see an increase, so there's definitely some kind of regulation going on with that in terms of replication. Okay, the last thing I want to address is that we talked about the structure of DNA, and basically DNA, can progressively get compacted because we know that there's lots of DNA, but it has to fit in a very small area of the nucleus. And so a typical chromosome may be hundreds of millions of base pairs long, maybe like a meter long. Think about a meter. But it has to fit in a cell that's 10 to 100 micrometers in diameter, for example. So we're going to have to kind of fold up this DNA and get it compacted into the nucleus. So a couple of things. So we're going to end up eventually, getting it into the shape of a chromosome. But before that, it's all this double helical DNA that's unwound. So a couple of things to think about too is that chromosomes, as we know them, are composed of what we call chromatin. Chromatin is really DNA and proteins in a complex. So DNA is not alone, but it's with protein complex. And the proteins are called histones. And they are very important. They are the first line of packing, first level of packing. Very important proteins, histones, very conserved, meaning most species have the same kinds of histones and sequence and whatnot. So it's like a universal protein, really important. And it's responsible for packing the DNA in the chromatin in the first way, the first level of packing. Here you have your several histones and the DNA wrapped around that, and then there's a little linker region, and there's the histones, the DNA wrapped around that. So each one of these is called a nucleosome, and it shortens the length of DNA by 7-fold. So it starts shortening the DNA, compacting the. So here you can kind of see them again, like I said, beads on a string kind of histone. The next level of packing is that you get this 30 nanometer fiber, they call it a solenoid or a zigzag. And so each nucleosome then like bends around. And so you form this kind of compact 30 nanometer structure. So instead of just you have a nucleosome, you have this nucleosomes all like kind of getting really, really close together to a 30 nanometer fiber, which shortens the DNA another 7-fold. After that, you have some looping going on. So after that, you form some loops. And the loop, you can see it on this picture. Here's some loop, 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 loop. Eventually, the loops continue to fold. Yeah, so there's some terms here. Euchromatin is not very compact. Heterochromatin is very compact. Okay, and so eventually, as you're getting to the chromosome shape, like what we see a metaphase chromosome, highly compacted, that would be heterochromatin. But euchromatin is like a little bit of the looped before it forms into the chromosome. So you can kind of see the progression, but of course you have like, you know, your double helix, then you have a nucleosome, and then you have this 30 nanometer fiber, and then you start forming loops. So it's still a little euchromatin, but then once you get really compacted and put into like a chromosome, then you have what we call heterochromatin. Okay, and so the idea is just to understand that DNA is tightly packed into a nucleus and into chromosomes, for example. However, think about it, if the DNA needs to replicate, you can't have it wrapped up so tightly. We learn that the DNA has to unravel, and then you go through all these steps of DNA replication. Likewise, we're going to learn about gene expression with Dr. Hayes. She's going to talk about how do genes get transcribed, for example. Well, we're going to see that the DNA has to change shape, has to unravel, also to express genes, do gene expression and transcription that way too. Histones themselves can undergo chemical changes so that they no longer wrap the DNA so well, and instead they'll help unravel the DNA a little bit. So stay tuned for that.